In this educational video, we will talk about the hormonal regulation of the menstrual cycle, as well as cyclical processes occurring at the hypothalamic pituitary axis, ovaries and uterus. Neuroendocrine regulation of menstrual cycle is a complex process involving 5 links: cerebral cortex, hypothalamus, pituitary, ovaries and uterus. The thyroid gland, adrenal glands and liver also take part in the regulation. The pituitary gland consists of an anterior adenohypophysis and a posterior neurohypophysis. The gonadotropin-releasing hormone regularly produced in the hypothalamus stimulates the production of FSH and LH in the pituitary gland, under the influence of which estrogen and progesterone are produced in the ovaries, and testosterone is produced in the testes. These hormones, in turn, suppress the synthesis of GnRH with a negative feedback. CRH, corticotropin-releasing hormone produced in the hypothalamus, stimulates the synthesis of ACTH, adrenocorticotropic hormone, in the pituitary gland, which leads to the production of corticosteroids by the adrenal glands. In the same way, TRH, thyrotropin-releasing hormone, produced by the hypothalamus, stimulates the synthesis of TTH, thyreotropic hormone, in the pituitary gland, which leads to the production of thyroid hormones by the thyroid gland. Corticosteroids and thyroid hormones also suppress the synthesis of CTH, corticotropin-releasing hormone, and TRH, thyrotropin-releasing hormone by the hypothalamus. The hormone SRH, somatotropin or somatoloberin, produced in the hypothalamus stimulates the production of SRH, somatotropin, by the pituitary gland. Dopamine produced in the hypothalamus has a depressing effect on prolactin synthesized in the pituitary gland. Prolactin ensures lactogenesis and inhibits the ovarian cycle by suppressing the production of FSH and GnRH. The hormone oxytocin is produced in the hypothalamus and passes to the posterior lobe of the pituitary gland. Oxytocin ensures lactation in the postpartum period and stimulates the contraction of smooth muscles in the myometrium. Antidiuretic hormone produced in the hypothalamus and transported to the posterior lobe of the pituitary gland regulates the production of urine in the kidneys. We will now discuss how these hormones regulate the complex processes that occur in the ovaries. But, firstly, we will briefly look at the structure and development of the ovum and its role in the formation of the hormonal function of the ovaries. The female germ cells of the embryo are called oogonia. At the 20th week of intrauterine life, there are 6 to 7 million oogonia in the fetal ovaries. In a newborn girl, the number of oogonia decreases to 1 to 2 million. The number of primordial follicles in the ovaries of a pubescent girl is about 400,000, of which 300 to 400 mature and ovulate. All the other follicles undergo physiological atresia. Now, we will talk about the structural and functional changes that the primordial follicle undergoes. During the menstrual cycle, maturation of the dominant follicle begins with the primordial follicle. A primordial follicle consists of a primary oocyte surrounded by a single layer of follicular squamous cells. At the beginning of the menstrual cycle, under the influence of local factors, a primary follicle forms from the primordial follicle. In the primary follicle, the flat granulosa cells become cuboidal, the oocyte increases in size. A thin glycoprotein membrane, called the zona pellucida, forms around the ovum. The primary follicle then develops into an early secondary follicle. This process takes place under the influence of FSH and LS. Under the influence of FSH, the amount of granulosa cells surrounding the oocyte increases, resulting in the epithelium surrounding the oocyte becoming multilayered. The main function of granulosa cells is the synthesis of estrogens. Granular cells are surrounded by Thecke cells. Under the influence of LH, the Thecke cells synthesize androgens from cholesterol. From the Thecke cells, androgens diffuse into granulosa cells, where Under the influence of FSH and aromatase enzyme, androgens are converted into estrogens. Estrogens stimulate endometrial growth and inhibit the production of GnRH, FSH, and LH. Further, the early secondary follicle turns into a late secondary follicle. As a result of the secretory activity of the granulosa cells, small cavities are formed, which are filled with follicular fluid. The fluid is full of estrogens. From the late secondary follicle, under the influence of FSH and LH, the early tertiary, then the late tertiary or Grothian follicle is formed. Small fluid-filled cavities merge to form a large cavity called antrum. Around the zona pellucida, a single layer of granulosa cells is formed, which is called the coronal corona. It has a protective and nutritional function. In the middle of the follicular phase, the estrogen levels begins to increase, which, on the one hand, continue to actively stimulate the endometrial growth, and on the other hand retain their suppressive effect on the hypothalamic pituitary system. This mechanism is necessary for the selection of a dominant follicle, the follicle with the highest number of granulosa cells, and FSH receptors will become dominant, which will help it survive in case of low FSH concentration. The remaining antral follicles will undergo atresia. At the end of the follicular phase, around day 13 to 14, the estrogen concentration reaches its maximum, and while estrogens previously had a depressing effect on the hypothalamic pituitary system, estrogens now begin to stimulate FSH and LH synthesis. Through this positive feedback mechanism, the FSH and LH concentrations in the blood increase, but the amount of LH exceeds the amount of FSH, because the Grothian follicle begins to produce inhibin B, which inhibits FSH synthesis. The peak increase in LH concentration stimulates ovulation. Ovulation occurs 10 to 12 hours after the LH peak. Under the influence of LH, the proteolytic enzymes plasmin, histamine, and collagenase flow out of the theca cells into the follicular fluid, causing the basal membrane of the follicle to rupture, which leads to the release of the egg from the follicle. Immediately after ovulation, the follicle is filled with blood and the hemorrhagic body is formed. Ovulation signals the end of the follicular phase and the beginning of the luteal phase, which begins with the formation of the corpus luteum. The formation of the corpus luteum directly depends on LH. After ovulation, granulosa cells under the influence of LH undergo luteinization. Luteinization is characterized by an increase in the size of granulosa cells and the formation of lipid inclusions, which determines the yellow color. The corpus luteum begins to produce progesterone, inhibin B, and estrogen. Progesterone suppresses GnRH synthesis, so GnRH, LH, and FSH levels gradually begin to decrease in the second phase of the ovarian cycle. Progesterone produced by the corpus luteum stimulates the endometrial secretory transformation, and inhibin B continues to suppress the synthesis of FSH. At the end of the luteal phase, in the absence of pregnancy, the corpus luteum begins to undergo atresia, causing the amount of progesterone, inhibin B, and estrogens to decrease and the suppressive effect of these hormones on the hypothalamic pituitary system disappear. As a result of all this, the level of GnRH and FSH start to rise again, and the cyclical changes resume. Next, we will talk about the uterine cycle, first of all presenting the structure of the endometrium, as well as the important morphological functional changes that occur in the endometrium throughout uterine cycle. The uterine cycle, endometrial structure. The structure of the human endometrium can be divided into two different morphological layers. The upper two-thirds is called the functional layer, the purpose of which is to prepare the uterus for embryo implantation. In the functional layer, proliferation, secretion, and regeneration of the endometrium take place. The lower one-third of the endometrium is the basal layer. Although the basal layer does not change during the menstrual cycle, it plays a crucial role in the recovery of the functional layer of the endometrium after menstruation. The structure of the endometrium differs depending on the stage it is in. At the beginning of the menstrual cycle, in the early proliferation period, the endometrium has a dense and relatively thinner composition than in the late period of proliferation. In the early period of proliferation, the endometrium consists of a stable basal layer and a very thin functional layer. The upper layer of the endometrium consists of superficial epithelium, which completely covers the uterine cavity. Some epithelial cells contain cilia. The maximum number of ciliated cells is observed during ovulation. In the own layer of the endometrium are fibroblast-like cells, which are called stromal cells. Endometrial epithelial cells form the endometrial glands, which are mostly located in the functional layer during the proliferation phase. Endometrial glands are long, curved tubular glands with openings into the uterine cavity. The endometrial glands are also located in the basal layer, which ensures endometrial regeneration after menstrual bleeding, as well as after endometrial curettage. After ovulation, the endometrial stroma undergoes decidual changes. The stromal cells become larger and rounder, with the appearance of glycogen in their cytoplasm. As a result of decidualization, the shape, structure, and function of the endometrium changes. As a result of all this, the endometrium turns into a secretory organ. The secretion produced by the endometrium contains glycosaminoglycans, glycoproteins, and glycogen. During the secretion phase, there is an accelerated growth of the spiral arteries, as well as an increase in the activity of lymphocytes and macrophages, which synthesize immunomodulators, cytokines, and various growth factors, which are essential for a successful embryo implantation. The uterine cycle, endometrial cyclic chain gesso. Let's briefly summarize the processes that take place in the uterus and ovaries under the influence of pituitary gonadotropins. From the first day of menstruation, which is considered the first day of the uterine cycle, 2/3 of the functional endometrium is shedded, and its complete desquamation ends on the third day of the cycle. Menstrual bleeding lasts on average of 3 to 4 days, and from the fourth day of the cycle, along with endometrial desquamation, regenerative processes begin. During these days, the follicles mature from the existing prenatal follicles. In the early days, the absence of a dominant follicle explains the low level of estrogens and the relatively high level of FSH. From the fifth day of the cycle, follicles that have reached the antral stage become sensitive to gonadotropins, so their rapid growth begins. At the same time, one of the follicles that have reached the antral stage is selected as the dominant follicle, and the growth of the rest is suppressed. From the 8th day of the cycle, the growth of the dominant follicle is accompanied by a significant increase in the level of estrogens, under the influence of which the proliferation processes are activated in the endometrium. The growth of glands, stroma, and blood vessels. The increase in the level of estrogens produced by the dominant follicle decrease the level of FSH by a negative feedback mechanism. LH level, on the contrary, begins to rise under the influence of both the direct positive relationship between estrogens and LH, and the high levels of gonadotropin-releasing hormone. On the 12th-13th day of the cycle, 12 hours before ovulation, the level of LH reaches its peak, and the size of the dominant follicle is 20 to 21 millimeters. Under the influence of FSH and LH, the proteolytic enzyme levels in the preovulatory follicle increase drastically, which causes thinning and destruction of the follicle wall, resulting in the release of the egg along with the surrounding granulosa cells. Ovulation, which occurs on average on the 14th day of the cycle, is accompanied by a sharp decrease in the gonadotropin levels and the formation of the corpus luteum, which is mainly under the influence of LH. The uterine processes are under the control of estrogens and progesterone. During this period, decidual-like changes occur in the stroma and glands, the activity of which reaches its maximum on days 20 to 21 of the menstrual cycle. Progesterone inhibits GnRH activity, which reduces gonadotropin secretion, and consequently the effects of FSH and LH on the corpus luteum. If by 24 to 27 days of the cycle, pregnancy is not registered, regression of the corpus luteum and progesterone reduction begins. A prolonged spasm begins in the endometrial vessels, followed by paretic dilatation of the vessels, increasing blood flow. The mechanical strength of the vessels is lost, the walls become brittle and rupture. Such a condition of the endometrium, when even before the rejection of the functional layer, bleeding occurs in the space between the functional and basal layers, is called anatomic menstruation and is detected several days before the clinical menstruation. As a result of anatomical menstruation, hypoxia, leukocyte infiltration and the activity of proteolytic enzymes increase in the endometrium, leading to desquamation of necrotic areas of the functional endometrial layer. A decrease in the level of E2 estrogens and PG progesterone in the ovaries leads to activation of the hypothalamus and pituitary gland, and an increase in the secretion of gonadotropin hormones, FSH and LH. After that, the cyclical changes resume again.